next presenter is Andrew Muir. Yeah, the mic is yours.
Okay, thanks everyone. Good morning. My name is Andrew Muir. Today I'm going to talk to you about some of our ongoing work to develop an objective, science-based framework for dealing with some important management of Coregonids that relates to restoration in the Great Lakes. So when we consider describing and organizing conservation units for species with high interest-specific diversity, two problems arise. The first is a science problem associated with recognizing and describing interest-specific diversity and then organizing that among conservation units. The second problem deals with operationalizing a plan to conserve and where lost, restore that diversity. So this slide shows the historical cisco complexes of each of the Great Lakes. If you consider what were named species and subspecies, as many as 10 forms, and I'm going to refer to them as forms, existed within a single lake. This slide shows the contemporary cisco complexes of the Great Lakes. And note that Lake Erie is devoid of any cisco populations. The deepwater forms of Lake Huron are thought to have intergressed into what we call a hybrid swarm. And in Lake Michigan, we lost 6 of the 8 forms that were historically present. Lake Significant, the north part of Lake Superior there, and then Lake Superior itself retained their historical cisco complexes, so that whole suite of diversity. So the objective of our work, and it is ongoing work, is to survey and apply conservation unit concepts to the problem of rehabilitation in the Great Lakes. So what we did was survey the literature, and we identified 7 different conservation unit concepts. The first, distinct population segments, is the primary basis for the U.S. Endangered Species Act. The evolutionarily significant unit, the second concept there, is sort of a subcategory of the DPS that applies specifically to Pacific Zone lines. Number 7, the designable unit, is the basis of the Species at Risk Act, SARA, in Canada. The other ones there, I'm going to define each of these as I go through the talk, but the other ones there are sort of lesser known concepts. So what we did for the purpose of this work, we're going to attempt to apply all of these different conservation unit concepts to each of the Great Lakes, but I can't obviously present that in 12 minutes. So what we did for this was apply those conservation unit concepts to Lake Michigan, where 2 remaining cisco forms exist today. The first is a shallow water form of Coregonid Symposium, the Sisko. It's I say shallow water, meaning that it spawns in near shore on shoals. And the second is a deep water form, Coregonid Symposium, or the bloater. And this location here in Traverse Bay is the known location where this shallow water form remains extant, the shallow water form of Coregonid. So today we're going to apply 4 of those 7 conservation unit concepts to the extant ciscos of Lake Michigan. So just to give you a sense for what we're doing, I chose to use the evolutionarily significant unit to give you a sense for how we apply these concepts to the populations. And I chose that because it's one of the more well-known concepts. So 2 criteria shown in the left-hand column here. We qualify a population or a population segment as an evolutionarily significant unit. The first is reproductive isolation. The second is that it's important in the evolutionary legacy of the species. So it's based on past evolutionary events. So what we did was we compiled in our survey a list of potential evidence that would support each of those criteria, and then we're in the process of compiling all of the available data supporting each of those pieces of evidence. So just to show you what we did, I'll run through the ESU concept here. So we have, with respect to the contemporary Artedi and Hoya in Lake Michigan, we have no data on movement of tagged fishes. So movement data could potentially support an assessment or evaluation of reproductive isolation. We do, however, know that there are differences between those 2 forms in spawning time. The shallow water spawning Artedi form spawns in 3 to 7 meters during November to December. By contrast, Hoya spawns in very deep water out to about 140 meters later in the season, in sort of December through to March. So we know there are differences both in spawning time and location for those two extant forms. So in that case, we would say we have positive evidence to support reproductive isolation. In terms of natural recolonization rates, we do know that in over a century, none of the forms that were extirpated from Lake Michigan, or any of the other Great Lakes for that matter, have re-expressed. So those forms have been lost, they're not re-expressing from the extant diversity today. We also know that that small population I showed you in Grand Traverse Bay that exists is not expanding at a rapid pace to recolonize the lake. That's debatable. I know some people will debate me. There is some evidence that it may be expanding and filling other areas of the lake, but it's certainly not to the extent that it historically was. And there are various reasons that others in this symposium will get into for why that may be the case. We do know that there's genetic differentiation between Hawaii and RTD. We can, unless I'm wrong, Wendy, assign individuals to either of those two forms using genetic methods. And we have no data on natural barrier efficacy. Mainly, it would be water depth as the primary barrier currently. Okay, so the second criteria here, we have ample historical and contemporary data showing that there is variation in phenotype and life history traits between those two forms. We also, as I mentioned, know that time of spawning differs between the two forms, so we can support that, that criteria with the evidence. And we have right now no data on adaptive diversity, so traits under selection from a genetic perspective. We have some ideas based on phenotype, form versus function relationships, but Trevor Krabenhoff to us here is going to present to you some genomic data for another lake, Lake Superior actually, so there's some evidence that there are areas of the genome that are under selection in these fishes, but for the case in Lake Michigan, we don't currently have data. So that's the idea. What we did was compile all of the existing data and then run it through this sort of ranking matrix and assess whether or not the extant forms would qualify as a different unit concept shown here, in this case ESU, and we saw that they most likely would qualify as ESUs in this case. The second concept I want to talk about is ecological and genetic exchangeability, because it's something that has not been formally embraced in sort of the conservation biology world, but it's an interesting concept. So it's set up as hypotheses. So a rejection of exchangeability would mean that the populations or forms would qualify as conservation units. Okay, so exchangeability arises from shared fundamental adaptations of a population or population segment, such as life history traits, ecological requirements, morphologies, ecosystem services, or ecosystem function, or demographic characteristics. So the idea is that if these things are exchangeable, they can be moved among different populations or among lakes. If they're non-exchangeable, as is the case for those two extant forms in Lake Michigan, Waiai and Otidi, they're not ecologically exchangeable. So therefore, they're separate conservation units. So I want to just explain this a little bit further here, because it's something that we haven't formally embraced in conservation biology in the Great Lakes. The idea that is presented in this Crandall paper, 2001 I believe it is, that if you take a look at this sort of idea from the perspective of rehabilitation or restoration planning, you have to ask the question of substitutability. Does a donor ecolog exist, and is the isolation from the potential donor ecolog recent? So what the authors mean by ecolog is that it's a unit that is ecologically or genetically exchangeable with other units. So we know, as I mentioned, for the case of the extant forms in Lake Michigan, they're not ecologs, they're not exchangeable. However, if we look to Lake Superior and Lake Nipigon, where we have that remaining extant diversity, some of those forms may be ecologically and or genetically exchangeable with other lakes, as is the case for Lake Michigan, where we're currently using Waiai, the deep water form from Lake Michigan, to repopulate the deep waters of Lake Ontario, in part on the assumption that the historical Waiai form in Lake Ontario, that's now extirpated, was an ecolog with the forms that are currently existing in Lake Michigan and other lakes. So the idea is intriguing if you can use it from a rehabilitation standpoint. The next concept I want to talk about here is the service providing unit. Again, this has been not well incorporated into conservation biology, at least in the Great Lakes. And the interesting part of this is that it focuses on functional ecology. So the authors, Luck et al. 2003, they use service providing and ecosystem function interchangeably here. And the idea is that they provide or might provide in the future a recognized ecosystem service. So this includes services to humans like fisheries, as well as ecosystem function. So if we take a look at this concept a little further and apply it to restoration ecology, this schematic here represents food web or habitat compartments in the Great Lakes. On the left-hand side of the slide are the nearshore waters, so less than 30 meters. The right-hand side are the offshore waters, pelagic and profundal compartments. And historically, in Lake Michigan, when we had 8 forms of ciscoes, deep water and shallow water ciscoes, these food web and habitat compartments were well linked. So animals spawned in nearshore, 3 to 7 meters of water. They would then move offshore into the pelagia to feed and also provide food for pelagic predators. We do know that the deep water forms undertake deal vertical migration, so they're moving up and down in the water column, linking those two food web compartments. We also know that things like the short-jaw cisco and potentially hoyide moved up and down the bank, linking the benthic production and carbon and nutrient cycling in those two zones. So the idea is that historically, these food web compartments were well linked. There's very strong functional cycling of energy and nutrients. This is a schematic of the Lake Michigan food web today. So it's a little bit of an over-exaggeration because we know that there are some non-native species that have filled some of those functional roles in terms of coupling environments, but in some cases those are entirely within a non-native food web. So we've lost some of that function. And again, this is a little bit of an over-exaggeration, but gives you the idea that understanding these things from a functional role in terms of linking habitat and linking food web compartments can be useful in terms of understanding some of that loss. And the last concept that I want to talk about briefly is the designable unit, and that's perhaps the most conservative and comprehensive concept. That's the foundation for the Canadian Species at Risk Act. And one of the primary pieces of this is qualifying adaptive variation and adaptive divergence. Typically, we think nowadays of genetics in terms of understanding areas of the genome under selection and adapted for particular exploitation of particular resources, habitats, or food. But what we can do is also look at form-function relationships. And this plot here shows delta 13 carbon on the y-axis. The less negative numbers at the top refer to a more literal carbon source. The more negative numbers at the bottom of the y-axis refer to a more pelagic carbon source. And this is plotted, this is Lake Superior ciscoes for the 4 forms of extant ciscoes in Lake Superior. Plotted here against body shape, and that's variants that come out of a morphometric analysis of body shape. The idea here is that there's a 0.22, so a moderate relationship between form in terms of body shape and function in terms of carbon use in this system. And given the biological variability in both of these kinds of data, this is actually a fairly good demonstration of form function. And Louise Chavarri is going to be presenting a whole bunch of these kinds of data for both lake trout and ciscoes later on in the conference. And so just to summarize, conservation units can provide a framework to inform rehabilitation alternatives. And the idea is you can attach risks to these kinds of data and scenarios to help inform that management planning. Inclusive concepts like adaptive evolutionary conservation, which I didn't talk about, and the DU concept are very conservative because they consider all kinds of pieces of evidence, trophic evidence, ecological evidence, genetic evidence, all of those kinds of things to make designations about forms for conservation and rehabilitation. Ecosystem function and services and adaptive variation, so genes under selection. You're going to hear a bit about that during this conference, or during this symposium, that are linked to morphology, and also the idea of exchangeability. These are all ideas that have not been well incorporated in conservation and rehabilitation planning, in part because they're new and some of the methods are new. And so I'll leave you with this. If the form meets the criteria for a conservation unit, which we're hoping to kind of assess through developing this paper, then it's ecologically and/or genetically distinct, which can help then inform management. So the idea is to build this framework, throw all the data we can at it, and see how these things sort of group out. And again, it's a work in progress, so I'll probably be talking to several of you here for data and information to help build this as we go. Thank you. There's some time for questions if anybody has them. Timo.
Thanks for interesting presentation. Have you considered any of the food web metrics, like on the historical food web versus the current food web, if you have quantified what has happened on the whole ecosystem? Do you think you have enough data on that, or have you considered?
Yeah, so that's a good question. The question is, do we have enough data from the historical food web that we can compare to the contemporary food web? And I don't know in terms of the lower food web that we do have a lot of data. What we do have are archived scale samples and some tissues from historical food webs that someone has looked at from a stable isotope point of view. There may be archived lower food web samples as well, but we haven't got into that at this point. Someone else may know if anyone's working on that kind of stuff in the Great Lakes, but I'm not aware of it. Okay, thank you. Oh, yeah.
So Andrew, under both the DU and the evolutionary significant unit are both operational under ID?
Are both operational?
Like you separated them because of jurisdictional, but your scheme or your plan is relevant to both.
Yeah, so the question is, are we going to consider one or the other, I think, of these kinds of definitions as we think about the ciscoes of the Great Lakes? And what we're trying to do here, the reason we surveyed all of those concepts is that the paper by Fraser and Bernachet, I think it's Adaptive Conservation Framework or something it's called, the idea there is that you don't need to either abandon historical approaches to defining conservation units. What you can do is use the concepts of all of those things to try and build a case. And so the idea here is that you integrate pieces of all those different concepts. And that's why the functional piece and the exchangeability piece is nice to kind of build in. So you can use all of that information to inform management options for Sisko. So we're not rigidly applying one definition or the other to try and say that this is